In this session, we discuss about pre-analytical variables, sampling, and harvesting. Sampling needs to be done under strict standard aseptic conditions. Samples are to be collected in sodium heparin green-topped vials. We should avoid clotting of the sample by gently inverting the tube 4 to 6 times. 1.5 to 2 ml of undiluted bone marrow aspirate from the first pull is preferred for fish. What we follow in PGIMER is we try to make at least 10 bone marrow aspirate slides from the first pull. Then the remaining sample goes for fish in a green-topped sodium heparin vial. This is done for diagnostic marrow. For follow-up cases, the first pull is always taken for flow cytometry. Always make extra smears of trephine biopsy imprints, especially in cases with difficult bone marrow aspirate, dry tap, or in cases with diluted bone marrow aspirates. Peripheral blood can be used if bone marrow is not available and there are sufficient number of abnormal cells in the circulation. If direct bone marrow aspirate or imprint slides are to be used, ensure they are fixed in Carnoy's fixative, preferably within 48 to 72 hours of aspiration for 25 to 30 minutes and kept in absolute alcohol, preferably at -20°C. Coming to sample transport and storage, sample is transported and kept at room temperature before processing. In extreme temperature conditions, it can be kept at 4°C. For culture, the sample is processed as soon as possible, whereas for harvest without culture, the sample is processed within 24 hours. Other standard and good laboratory practices need to be followed in all the cases. At least 2 patient identifiers, identification of type of sample like peripheral blood, bone marrow aspirate, FNEC sample, or any other body fluids. Note about the time of collection, clinical diagnosis. All these need to be documented in all the cases. Now we proceed to the demonstration of the steps involved in harvesting. Thank you.